Our last presentation before our lunch break is to be given by Dr. Claude Gingras. She's a primary investigator at Samuel Lunenfeld Research Institute, Department of Medical Genetics and Microbiology with affiliation to Mount Sinai Hospital at Toronto. Dr. Gingras comes with prior experience at the Institute of Systems Biology, Seattle, and her research interests span cellular mechanisms involved in cancer and also mechanistic aspects of drug resistance in cancer drugs. So it's a pleasure to have you here. Go ahead.
Well, I'll start by really thanking Rob and Mark Chance and Gary for a very, very nice opportunity to be here and present some of our recent work. Is that working? This is great. All right, so what I thought I would do today, it's going to be sounding a little bit crazy and all over the place, but I want to give you a little bit of a view of what we're doing in the lab and in collaboration with several other groups. And most of the work that we're talking about today is going to be with chartering kinase and phosphatase interaction network, or Interactome. And you may ask, why do we care about that? And one of the reasons we care about that is because those signaling molecules control essentially every aspect of life and almost every aspect of cellular regulation as well. So they do that in part by establishing interaction, either a stable interaction or a more transient substrate-enzyme relationship type of interaction with different proteins, including regulatory protein, target proteins. But although they have been studied for many, many years, they've largely escaped detection, at least in yeast, in the high-throughput studies that have been made. So we decided to revisit this, and the first part of my talk is going to be dealing with a investigation, systematic reinvestigation of kinase and phosphatase interaction network in Cerevisiae. And if I have a little bit of time, I'll tell you a little bit more at the end about our efforts in my lab to look at mammalian interaction networks. So one of the reasons why we thought it was a good time now to reinvestigate yeast interactome for kinase and phosphatases is because the technology has improved a lot. First of all, we have mass spectrometers today that are much more powerful than what was available when the work of the other, and Claude Gavin was published in 2002 and in 2006, and the work that Jack Greenblatt and Evan Krogan did in Toronto. And we knew that because we were facing the same thing in our hands when we were looking at our interaction network 5 years ago and the interaction networks that we are looking at now. But one very, very important point for kinases and phosphatases is very often they do not form a very stable complex that withstand the two-step of purification that these studies in yeast were using, which is a tap purification. So, and this we knew, again, from our work where we were looking at tap or tandem affinity purification. I'll come back to that in a second, for a serine threonine phosphatase called PP4. And back in 2005, we were really, really proud that we had discovered new interactors for this phosphatase that could explain some of its role in DNA damage signaling. But what I just want to stress is this is actually data from our lab now, and all we've done, this is the same instrument, this is a LTQ work, and this is now done with a different tag on the same instrument, and you can see that my nice, beautiful complex, which I was so proud of, is now just way here at the bottom, and there is all this new and unexplored interaction network and number of interactors that we have there. And what we know now, and I will not go into details about that, is many of them are actually now bona fide substrates for our phosphatases. So we thought it was actually a good time to look at that a bit more systematically. And just to refresh people's memory, tap purification works great. Essentially what you do in this case is you will put on your gene of interest, you will fuse a bipartite tag, which comprises of two moieties that are each used sequentially for affinity purification. And you have a dual purification step. So in the way that was used for the yeast data as well as our mammalian data, the first tag, or the most distal tag from your gene, is actually protein A, which binds really nicely to IgG spheros or magnetic beads with IgG. And this is separated by from a calmodulin binding peptide by this little red sequence here that bears a recognition site for a protease. So the purification scheme goes as follows. You purify on IgG cephorose, you clip this little tag, then repurify on calmodulin cephorose. And this can all be done under fairly gentle conditions. And at the end, what you end up with is a very nice, purified, fairly stable protein complex, or rather a mixture of protein complex. By contrast, the flag purification that we've been using recently has been employing a very simple one flag or 3 flag tagging of your protein of interest and a rapid one-step purification. That leads to the recovery of your protein of interest, but of course there's much more background associated to that. And again, just to remind people, TAP is really good for a stable complex, and this is what has been published by the other group. The background of flag, especially in mammalian cells, is very high. In mammalian cell, we have greater than 200 protein in any given run that are actually background contaminant. But the good thing is, and that was really clear from Mike's talk this morning, is this background is extremely reproducible. So if you do the experiment over and over again, you have proper controls, and you do quantitative proteomics, you can actually interpret the data. And again, this will be stressed more this afternoon as well, but Mike did a great job at introducing. If you combine this kind of approach with statistical tools and interaction database, you can actually get at the answer. And this is what we decided to do. And again, we're looking at enzymes, kinase, phosphatases. We want to get at their target. So for us, there was really no other way to go about that. So for the yeast project, we first decided to use all of these new knowledge that we had to rebuild our pipeline for the analysis of the interaction. And so the first thing we did is work on the sensitive experimental platform. And in our case, what we decided to go with, and this is actually the work of many, many talented people, mostly in Mike Tyer's lab for this experimental platform, we went with protein that are epitope tag and that have induction made possible by a GAL-inducible promoter. So we can have a biological material here that is a yeast strain that is expressing a single protein under the control of a GAL promoter. We do short induction so that we do not get a massive overexpression, and we did nice titration of this. Following the lysis of the cells, we can rapidly capture the tag protein on magnetic beads, and we perform relatively quick washes. And so that is our affinity purification step here. And this actually is fairly quick. It takes us less than 2 hours to go from this sample to the digestion, which is now done on beads. So we're not even trying to elute the samples and then perform the digest. We add trypsin directly on beads. And this is now simply submitted to gel-free liquid chromatography and mass spectrometry. And for most of this project, we've been using a LTQ instrument. We have done some runs on the Orbitrap mass spectrometer, but most of the work is simple LTQ. So then we got stuck because we had all of this very efficient pipeline. We can make a lot of samples. And we really wanted to make sure that we could handle the data. And the next couple of slides, I just want to show you our strategy and essentially our solution to that. And again, average flag run, it identifies many proteins. Most of them are contaminant. And so how do you actually keep track of all of your samples? Are you going to do that on Excel sheet? If you change the protocol a little bit, how are you going to be able to recover things? And then I'm at the Samuel Lunenfeld Research Institute, and we were facing we had many projects facing exactly the same thing. So we have multiple labs, we have multiple mass spectrometers, so very soon we'll have 15 of them. And when I say multiple labs, we have 10 labs that are actively involved in mass spectrometry. We have about 40 trainees, in addition of the personnel of the mass spectrometry facility, running their own samples, and about 100 to 150 people analyzing their own data. So we need something that will be, at the same time, very powerful in terms of keeping the data, but very easy for these people to analyze, and very flexible to pipelines. And so we develop, and again, this was work initiated by Mike Tyers, we developed a very efficient pipeline for the data processing and the analysis. And this is just a LIMS system, essentially. But it's a LIMS system that we devise for this kind of workflow. And it's comprised of 2 modules. We call it Prohits. And we're going to, it's made to be open source. We are testing it right now. So if anybody is interested, let us know. I know Rob will give it a try. So it's comprised of 2 modules. One of them is dealing with our data management from the mass spectrometer. We have scheduled backup files, scheduled search with user-defined parameters. But more importantly for interaction proteomics, we have this module called the analyst, in which the data is organized in a project-specific, bait-specific, experiment-specific, and sample-specific manner. And with a lot of annotation at each of the steps, so that once you actually link your raw file to your sample, you can actually really know what you've done to get there. So several years later, when the student is gone and you're trying to figure out, oh, what was that run, you can actually track it down. And so I just want to show you a couple of pages from it. It's fairly massive in terms of the number of functions that it has. But this is actually a very important page as far as a PI, a boss, is concerned. And this is where the people annotate their experiments. So every time and again, I was showing you, oops, sorry, I'm going the wrong way again. I was showing you where we are looking at biological material, affinity purification, peptide preparation, and gel-free LCMS. So we have places in the database to annotate every one of those experiments. So we have text-based protocols that are available via drop-down menu to input those biological material, affinity purification, and so on information, so that you can view them, you can modify them, they get date stamp, so that once you have them enter, we can always track them back. We have also another area where we can select control vocabulary, which is something that the administrator of the project will say, I really, really want you guys to put the exact cell line that you've been using, so that then you can structure your data and do several other operations with controlled vocabulary. And of course, you have places for additional detail and Western blot image and so on. So this is actually really good. So all of the data that we have for the yeast project, we have 1,500 different immunoprecipitation coupled to mass spectrometry experiment. Each single one of them is annotated like this in the database. For the other project, we have like thousands of experiments, for example, in mammalian cells, where we have this annotation as well, which is really useful. What it is also good, we've spent a lot of time to try to develop graphical user interface that are fairly intuitive to biologists and people that are looking at the data. And this is one of them, which is what we call the comparison page, that allows us to align in this kind of column axis. You have, this is a control run, and you have a number of different purification experiments. And on the row axis is actually, it goes way down, of course. This is all of the hits, or the identified protein. And it's essentially color-coded in this case by spectral count. You could have different color coding based on different properties that you're looking at as well. Everything can be toggled at. And this list here that I'm showing you is a filtered list in which proteins that are actually on the background list that is user-defined have been removed simply for sake of visualization. Of course, there are links to interaction database. In this case, we're displaying interactions that are already known and interactions that are not known in the BioGrid database, and also other links. You can also view your peptides. I will not show you that, but we have a button to directly map this network onto a cytoscape network, which still keeps all of the annotation shown here in dark blue as an edge attribute in cytoscape. So the goal of this entire project was, and this is just kind of an unfiltered list that you would get. So the goal of this entire project is actually to be able to have a pipeline that can be exported using different lab, and just is a very robust way to keep the data alongside the experimental node. All right, so now we have an experimental platform. We have a way to actually record the data, and now we need the smart people to help us figure out this data. And you will hear more about that from Alexi this afternoon. What we're generating, I told you we did about 1,500 experiments, about 500 of them we decided to include in this Kinome network. And so what we did is we sent a hit scoring metrics, which is essentially what I've shown you in the comparison page. So we have all of the base and then all of the hits, and we've been sending all of the parameters, including spectral count, peptide count, and coverage, and so on. And then we just have been sending all of that to Alexi. And what Alexi has done, and he's going to talk to you more about that, is something very much in line with what Mike has talked about this morning, is really try to get a better idea of the real positives and the true positives in a sample in a sea of mostly negative interaction. And what I mean by that is that when we sent the metrics to Alexi, there was 38,000 interaction in this matrix. And of course, most of them were only background contaminants. So Alexi has been able to modelize the frequency or the distribution of each of the hits across all of the purification. And this has allowed him to put p-values on our interaction. So after he's done that, and again, he's going to talk to you about that this afternoon, he sent us back the scores, and then you can reorganize the score in a different way and analyze, do some kind of network analysis, either simple clustering type of approaches or more complex follow-up studies as well. So just to give you an idea what kind of data, and I don't know how well that goes, so the statistical tool is called SAINT for significance analysis of the interactome. And this was actually the spectral count matrix prior removal of the contaminant with SAINT, and this is what happens after. So most of these contaminants were removed. So from our 38,000 raw interaction, we distilled a network of 1,800 roughly interaction with about a sixth of the yeast genome. So this is an interaction network for 125 kinases and 35 phosphatase, and I'm not expecting you to be able to see any of that. The only thing that I want to show you is this is actually just network from our own data set. It actually pretty much doubles all of the interaction that were known for kinases and phosphatases in the literature, both in small scale studies and in high throughput studies. There is not much that you need to know, except that everything that I show by a big orange node or circle here is actually a kinase catalytic subunit, and the greenish blue nodes are phosphatase subunits. And the size of the nodes is actually proportional to the connectivity, how many neighbors this particular protein had in our own network. So the first thing that we did is we started making sure that we were not completely crazy, and a poor, I actually forgot her name there, is very important, Laurie Boucher, has spent a lot of time validating by IP Western many of these interactions. I'm just showing you a few. And so we know that we're dealing with something that we can validate, we can reproduce, we think is a good data set. And I just want to make a couple of points on what is in that data set. And the first thing that we were able to observe, because it was quite striking, is there is a huge enrichment for kinase-kinase interaction. That is, if you're a kinase, you're much more likely to interact with another kinase than you are to interact with any other protein or classes of protein within the cell. And this is just a plot of those interactions that we have observed. I don't really know what it means. Initially, it scared me a lot because I thought maybe we were there were kinases sharing peptides, but we did a lot of annotation. One of the things that we think it may mean is we already have in evidence from many different small-scale studies that many kinases are organized by scaffold into signaling cascades. And we think that at least some of what we're looking here might be that, so that you have either a kinase-substrate relationship with kinases or something even mediated by scaffold. So this is something that we're going to continue working on. What I want to spend a couple of minutes on now is a very cool finding that we had. So there was a lot of information in this network, and it was hard to figure out for us what we were going to follow with the biology, because it's not that you can just recapitulate the interaction, but you have to figure out what it means at some point. And we're just beginning to do that. So this is actually a bait clustering. So this is all of our baits organized. And this is actually a profile that Alexi has done actually that look at how similar the IPs of all the baits are. And it can cluster things into little areas of locally dense interaction or baits that look very much like other baits. So we spent a little bit of time in the paper, which I won't go into details here, to look into some of those clusters. The one I want to draw your attention on is this one here, which surrounds target of rapamycin. And of course, target of rapamycin is dear to my heart because I spent 8 years in my PhD working hard on understanding this, essentially. So this is actually a slide stolen from my own PhD, where TOR stands for target of rapamycin. So it's a very, very important molecule in the cell because it integrates the nutrient, the response to nutrient with downstream action. So of course, I was in a translational control lab, so we were looking at translation factor, which are really well characterized as downstream target of TOR. But you have to know that rapamycin is used right now in the clinic to prevent graft rejection in transplant patients because it has a very important role in the immune system, but also that TOR inhibitors are used in the clinic as well as anti-cancer agent, because what I didn't show on this slide is that pretty much every molecule, every kinase, every other molecule that leads to TOR activation is either an oncogene or a tumor suppressor. So perturbing this pathway actually has huge impact on the cell. So the reason why we got really interested in our TOR data is we found many, many new things. TOR had completely escaped all of the high throughput data set in yeast. It was nowhere. And so just to be a very, very simplistic view of what the TOR pathway looks in yeast downstream of TOR, again, TOR is a kinase. There is two of them in yeast called TOR1 and TOR2. One of the complexes that they form is called TORC1 complex or TOR complex 1 with other proteins that mediate some of its function. And this complex control many different things. So everything in green at the bottom is a transcription factor. So there is a regulatory network that goes from the nutrient. In yeast, it's mostly nitrogen, carbon, and also phosphate, and controls the activity of those transcription factor. It controls the re-entry of nutria via this other kinase called NPR1, which is a nitrogen permease regulatory, and via some phosphatase regulation here. There is this nice area here, which I will come back in a few seconds, which is implicated in retrograde signaling. And this is actually a feedback mechanism between the mitochondria and the nucleus to say, like, well, do I have enough nutrient? No, make more, get more in, and so on. So this is a really important process. So all the arrows and lines that I'm putting here, except for this complex here and this interaction, they're genetic links. There was nothing known about physical interaction within this network. When we look at our network, this is what actually we saw. And I'm not going to ask you to go into more detail, but into any detail on that, but this is the TORC1 complex in this area. These are all of the interactors that we have. Everything that's a red arrow is coming solely from our data, and the thickness of the arrow in this case is proportional to how much peptides we have, so spectral counting kind of. And so what we saw is we saw TORC1 nicely linked to PP2A family phosphatase, which was really known genetically, but this is actually the first evidence for that physically. It's really linked to ammonium metabolism, which is, again, important in TOR signaling. And what we were really interested in is we found 4 kinases that were physically associated with TOR and had never been shown to be associated. And we could validate these. So this is another of those co-IP. So we can IP each of the kinases and blot against either TOR1 or TOR2 and see that in all the cases they bind TOR1 and most of them also bind TOR2. So what are those kinases? Well, one of them was NPR. It's that kinase that was genetically known to be implicated in TOR signaling via tryptophan permease turnover. So this is what NPR1 interaction network looks like. What is really interesting, first of all, in addition to bind to all components of TORC1, it actually physically interacts with that ligase called RSP5. And this is the one that actually antagonizes the effect of NPR1 on the tryptophan permease. So you have those 2 molecules that play antagonistic role on the same substrate. Now they're physically interacting with each other. Not only that, but NPR1 was shown, we've shown that it interacts physically with the AMP kinase. So that's probably an integration of the amino acid signaling to the carbon signaling mechanism, which is also completely novel. And we have even a binding of NPR1, which I believe I showed here in both of these cases. We have a binding of NPR1 with GCN2. GCN2 is a very important kinase, both in yeast and in mammalian cells, and it controls translation via phosphorylation and inhibition of a translation factor. Genetically, these were all known to be implicated somehow in TOR signaling, but again, this is the first map, physical map of this network, and it offers actually a lot of views, a lot of avenues to go further into the understanding of this pathway. I just want to say a couple of brief words about retrograde signaling. I already told you it's essentially a feedback mechanism from the mitochondrium to the nucleus and back, and it's really controlled by different molecules that sense the level of glutamine and glutamate, which are the main nutrient for yeast as far as main source of nitrogen. And again, this is a map mostly done with genetic only data. And this is actually our physical map of this network. So proteins that are very important in this complex, we actually find that they actually physically associate as well, so which was also very new. So I told you we were identifying 4 novel kinases implicated in TOR signaling, and that we are interested in pursuing them. So this is the first step of what we do, and I promised Jeff that I would show some yeast stuff. So this is one of those new kinases called FMP48 that was only assigned to be, it was identified by one of those shotgun proteomics experiments to be localized probably in the mitochondria. So FMP48 had several interactors, again, the TOR complex, PP1 of protein phosphatase 1 complex, proteins that are involved in different aspects of the cell growth. But what was really interesting to us is it also physically interacted with that protein called MKS1 that's really important in the retrograde signaling pathway. And so from this network, we started asking question, well, it looks like it could be involved into some nutrient sensing. It looks like it could be involved in some target of rapamycin signaling. And so we started doing some experiments. And one of them was simply to try to grow the yeast overexpressing mild amount of FMP48 on different carbon source. And as you can see here, the cells that express FMP48 grow fine on regular source, such as raffinose, but if you switch them to a different poorer medium, they're almost completely unable to grow, implicating their role in carbon metabolism. And you can do the same thing, but then look at sensitivity to rapamycin. And in fact, they are resistant to rapamycin, which really implicate that they have this has a dominant effect downstream of TOR signaling. If you look at those cells and you look at the mitochondrial morphology, and this is really hard to see, but all you can see is those are different. And this is actually a marker for mitochondrial morphology. So we're really perturbing the mitochondria under some conditions. And this is actually just a microarray experiment that compares those FMP48 cells with cells overexpressing that retrograde components. And you can see that there are classes of protein that are co-regulated in one way, but then there are huge classes of protein that are very specific to one or the other pathway. So we're really interested in following up those proteins. Lastly, the other one that we were looking at, the other kinase, again, a novel kinase that actually was on one of Jeff's slides. We renamed it as NNK1. NNK1 was really interesting to us because all of its interactors were in the nitrogen sensing pathway. More importantly, it interacted with URE2, which is a known player in the GLIN3 signaling. And so what we did in this case is first we check again that what was the sensitivity of the cell to rapamycin as opposed to FMP48. Overexpression of NNK1 actually renders cells hypersensitive to rapamycin. And what it seems to be doing, it changes the localization of GLIN3, which is this transcription factor that's really important for the upregulation of nutrient responsive gene. And you can see that very nicely if you look at a microarray experiment with GAL1 and NK1 versus a vector. All of the proteins that are all of the genes that are upregulated are known targets for the nutrient sensing pathway. So this really, taken together, this really helps us to redefine the TOR pathway and to start putting some physical interaction. So our kinases, NPR1, NNK1, and FMP48 now fit in different parts of the pathway. But what I was not able to draw on there is actually whether they act as direct kinase for some of these components. And this is what we're doing right now in that process. So I just want to give you a couple of last notes on the yeast genome. I showed you that we've been able to demonstrate, I think fairly well, that nutrient signaling molecule are physically linked in the cell. So once you have a method that's sensitive enough, you are actually able to find physical interaction that had otherwise escaped detection. What I didn't have time to talk to you about is that the two biggest hubs in our network are actually serine phosphatases, are actually phosphatases. GLIC7 is a serine phosphatases, and CDC14 is a dual specificity phosphatase. And what they seem to be doing in our network is integrate different pathways. I told you that kinases interact a lot with other kinases, and this leads, really opens the avenue for the identification of new signaling pathway. What I haven't shown, because we don't have the data, is we haven't even started probing condition-specific interaction for this project. We haven't been able really to order pathways, and we still don't think we're the most sensitive ever. So I think there is a lot of improvement to be done. So I'm going to stop there for the Kinome project contribution. This was a big team effort that involved three labs, my lab, Alexi's lab, and Mike Tyer's lab, and many people. So the work that I've shown on TOR signaling, it was all done by Jeff Sharon. Laurie has done all of the validation of the interaction. Zen, which is a technician for me, has done 1,500 IP mass spec experiment, and other people have contributed other parts that I didn't really describe in this. I just want to switch for a few minutes just to give you a little bit of a view of the rest of what we're doing in my lab, and that has a lot to do with serine threonine phosphatases in human cells. And I just want to bring this picture to remind people that serine threonine phosphatases acquire specificity, target subcellular localization, and activation cue via assembly with other protein into supramolecular complexes. And really, to understand how they work, you need to understand what the complexes are. And this is just an idea to show how complex these things are. And this was even before we started doing anything. There was many proteins that were known to be assembling as holoenzyme with PP2A. And essentially, the goal of what you're trying to do is to reconstitute specific holoenzyme to specific substrate and really know this connection. And so we're not there. And I just want to give you a quick update of our PP2A interaction network and where we're going with that, and the kind of leads that this gives us. And this is just a little snapshot because I'm running out of time. So this is our cytoscape map of the data that we have for the PP2A interaction network, and this is now several months old, so the background color has changed now and we have more interactions. But essentially, this is where the PP2A, there's two catalytic subunits, this is where they stand. This is one of the known binding partner, also called the scaffolding subunit, and all of the protein in blue are known binding partner as well, known as regulatory subunit. So you can see that we have a nice complex. So everything that has a square is a protein that we've used as a bait in these kind of experiments. So of course, there is a very high density of interaction there, and that's exactly where we're going to go for the rest of the talk. Of course, we are merging into different interaction networks that the lab has made. So now the entire lab data set is quite big. And this is, again, mostly work from my postdoc, Michael Mullen, and also other people. What I want to draw your attention on is this thing here, which is a new complex that we just published, but I want to show you some new data on that. So we published this this year it's a work that was done by several people in my lab. And what this complex is PP2A catalytic, it's scaffolding subunit, a known regulatory molecule, a known interactor of all that. But all you can see here is it's very, very well connected. In fact, there is only a couple of places where there is no interaction. What is interesting to us is actually this is a kinase from this T20 family of serine threonine kinase that actually physically associates pretty stably with our phosphatase complex. Together with this kinase is this protein called CCM3. Now, CCM3 is really interesting for neurosurgeon because it's one of the 3 proteins that is mutated in cerebral cavernous malformation, which is a kind of angioma. So cerebral cavernous malformation are fairly frequent. They are present in 0.5 to 1% of the population. Of course, for most people, they're asymptomatic. You will have one of those malformations, which is essentially a mesh of capillaries that have lost their integrity and tend to bleed a little bit. But in people with familial cases, this is actually quite bad because they get many of those lesions. The lesions are really, really big. And the range of phenotypes and clinical outcome is quite bad, especially if people have mutation in CCM3. So it's thought to be working as a two-hit kind of it's an autosomal dominant disease, but all of the mutations that have been characterized so far are loss of function. So I told you there are 3 genes that are implicated in this disease, and when we started our work, it was thought that the 3 of them would actually form the same physical complex. And the only thing that I want you to bring out from this is we looked at CCM1 and CCM2, which are the 2 other genes, and as you can see, they interact really well with each other and with that other protein. This is a CCM2, but it did not really overlap with our complex that we had just identified, which has CCM3 in there. So we're dealing with protein that have the same clinical outcome in terms of gene that have the same clinical outcome, but are really in 2 different complex. And I'm going to stop there with this story because I'd like to take some question, but we're actually moving forward in the biology of that and maybe some regulation of the phosphorylation and signaling. So what I want to say about this and about the different complex is by taking a non-biased approach and disease gene, we actually have learned much more about their mechanism and their function than all of the other approaches that other groups had taken, that were not involved, that were doing more directed. So you think CCM3 is going to be interacting with P53, you bind the antibody to P53, you do an IP, it doesn't bind, you go to the next. So I really think that even in the case of disease gene, it really is a good occasion to look at it in an unbiased way. So the work that I didn't really present to you from my lab today has been done by a group of people. Michelle Keen is actually our lead on the CCM story now. All of the work that I've presented today has been done at the SLRI by a very, very talented group. Brett Larson is our mass spectrometry coordinator, so he's doing a lot of great work in trying to develop better technology to more sensitive approaches and so on. The Prohits software has been developed by 2 programmers, Frank Liu and JP Zhang, and we have now a co-op student that has joined the team as well. The mass spec group is in theory led by me, but nobody really leads those guys because they're so talented. So it's Mike Tyers, Tony Posson, Jeff Renna, Jim Woodgate, and Jim Dennis. The CCM work is done in collaboration with a group in Toronto, and I'd like to thank all of these people. You will hear more about these things this afternoon a bit, and the funding agencies as well. And thank you very much for your attention.
Thank you for the presentation. Excellent talk. We have time for a couple of questions from the audience. Yes, please.
You mentioned that the kinases tend to attract each other. Have you looked at anything in the molecular structure of kinases that would lead to that?
So it's a great question. So is there anything specific about the kinase domain that would? So we've talked to actually this guy here, Frank Sicari. He's a structural biologist, and he lives for crystallization of kinases. So we haven't been able to find anything obvious in the so we were looking. So many kinases will dimerize, but they normally do not dimerize via the kinase domain. It's usually something outside of the kinase domain. Because of the number and the type of interaction that we get, we haven't been able to find any rules for which type. We haven't looked. It looks like we have many kinases that interact either intra-family or inter-families as well, if you group the kinase into different structural domain. And we haven't been able to find any simple rules to that. What we have been able to find is a mild enrichment, and I don't think it's really statistically significant even, for proteins kinases involved in the same pathways to interact with each other. But even then, the statistic was not complete. I mean, the number of data points, I think, is not that high to start with. So, yeah, Rob.
Are you able to distinguish between what you think are the substrates of the kinase and phosphatases from those more stable interactions?
Right. With the yeast data set, I would say no, because we don't have enough. I don't think we have enough coverage. With the mammalian data set, and because we also had prior, we had an entire network that was tandem affinity purification, that gave us really the core component of our mammalian data set. So in this case, pretty much yes. What I didn't show, and I'm not even going to flip the slides back, is we have been doing kind of systematic RNAi treatment or treatment with phosphatase inhibitor and try to look within a complex that you pull down, how many of them, we can map any phosphorylation site, how many of them look like they would be substrate which phosphorylation is changing. And for the mammalian data set, it is a majority of our interactors appear to be regulated by phosphorylation. Most likely, I don't know if it's going to be in cis or in trans within the same molecular complex, but they seem to be substrates. And it's really a majority. But for the yeast data, I have no idea. Please, a question in the back.
Dr. Pandey.
Particularly all the yeast to hybrid type data, would you venture to say that many of these kinase interactions that you're finding are actually indirect and not direct? I know that you suggested in passing that they're more likely to be direct, but there are a lot of studies that have been done, including substrate type of studies, but definitely if the interaction is outside of the kinase domain, we're not looking for transient interaction, that they should have been captured and well represented in the yeast to hybrid data set.
That's true, and actually we do not have a great, so even for the yeast to hybrid data set, the number of interactions that actually involve kinases were actually pretty low. So in the data that was published and filtered with whatever criterion that was decided, the data was pretty low. But I know a certain Pascal from the Vidal lab that really wants to get his hands on our data set and re-interrogate their newer data set for yeast. We're doing something very similar for the mammalian networks that we're building, and we're looking at doing systematic comparison of binary type of approaches with our data here, because I think we're capturing kind of a mix of direct interaction. Most of them are still indirect in our network, but I think we are going to be able to expand a bit more as compared to what was done with the TAP data. But yeah, that's a great question. There is one in the back.
So you said about the kinases being in a linear pattern. The reason for it to interact. But what about phosphatases? Do you find a pattern that they would regulate the kinases, or they're just come in and go to the system?
So phosphatases, especially CDC14 and GLIC7, CDC14 interacted with many kinases. I can't remember. I think the number of kinases it's interacted with was 28 in our network, which is why we systematically tested all of them by immunoprecipitation in Western, because we were really concerned initially. There were patterns for CDC14, but patterns more in terms of function instead of classes of kinases it bound to. So it did not seem to bind to a specific family of kinases. In fact, it was spread across all of the tree of the kinase alignment, but it really seemed enriched with nuclear type of which you expect, of course, for nuclear phosphatase. And so for GLIC7 was also something very similar. So there was an enrichment, of course. GLIC7 is protein phosphatase 1, and it is known to bind to protein via a short linear motif. So of course, in the interactors of GLIC7, many of them had this motif, but not all of them. So we're dealing, again, with both direct and indirect interaction, but there was an enrichment there and a bias for things with that linear motif.
Gaurav?
You mentioned that you compared your network with literature to see if any of the interactions are novel.
Right.
What type of method do you use to confirm whether that has not been found in literature? Do you test for each interaction?
No, so you mean like, did we test all of our novel interactions, or did we curate the literature?
Right.
So the literature curation has been, so we've made a big effort. So Mike Tyers also has BioGrid, right? So people that are the curator of BioGrid for the last year of the Kinome project have systematically made sure that they were up to date with the curation of all of the yeast, kinase, and phosphatase in terms of their interaction, so that when we actually compare the data set, it doesn't mean we didn't miss any, but I think we have done a pretty decent effort at populating BioGrid with the literature data. So what we've done actually is a lot of text meaning using different tools and just populating the BioGrid database with that, so that when we actually started doing the comparison, and we did different ways to compare our network with the known network. So part of it is for some of the figures, if we were letting all of the high throughput data in terms of like doing the maps and the visualization, this was getting really too crowded. So what we've done is, for display purposes and for some of the calculation, we've looked at experiments that were reported by at least 2 independent studies or 2 methods and so on. And that has really allowed us to look at that. But when I say that we have approximately double, this was even with raw interactions, like even with 1 report, as per our annotation of BioGrid.
Thank you. Thank you, everybody. I
Thank
think we'll call that our morning session. Let's thank our last speaker again.